Thank you, Dr. Yapi, for the warm introduction. Greetings, everyone. My name is Jason Redding, and as it was mentioned, I'm a geneticist at South Africa, at the South African Stud Book. My presentation is on using modern genomic markers in selecting healthier livestock. I didn't really get a brief on what to present, so I looked at the agenda for today's meeting and saw we were talking about resilience and fertility. So that's what I'll be talking about today. So, what is genomics? Well, genomics is the analysis of the complete set of genetic information. We are not just looking at the hereditary part of the animal, who is the parent, what gene was passed on, but we can get all the genetic information from that animal. We use single nucleotide polymorphisms, and these are used to increase accuracy on parentage verification and discovery. It's currently the most accurate form of parentage verification. The discovery part comes from the multiple sires. If you don't know who the sire is, we can identify the sire genomically, and it is so accurate that we are also able to determine the parentage between twin rams for a progeny, which we have successfully done and implemented. Most of you know about the genomically enhanced breeding values, increasing your prediction accuracy on your breeding values to select your genetically superior animals. But today I'll be talking about the single gene traits. These are all available on the newest version of the SNP chips that we offer through the genomic service at SA Stud Book. And we have verified all of these SNPs either through the online Mendelian Inheritance of Animals website or the Ensemble website. And we do have a good relationship with Weatherby Scientific in order to validate a lot of the ovine mutations that I'll be discussing. The majority of these single genes work qualitatively, that meaning that they are not influenced by the environment, and it is a single set of genes. It is a single marker that influences a phenotype. Majority of you know the easy one is polled horned. It's a single mutation. It is not influenced by the environment, and it dictates the phenotype of the animal. So first, we'll start with fecundity genes. These are very well known in the sheep breeds. The first one discovered was the Borula gene in the Borula merino in Australia, and it was observed to have an additive gene effect, meaning that an animal that was a heterozygote ovulated on average a little bit more than the wild type, while the animal that was homozygote, meaning it carried 2 copies of the Borula gene, had a plus more effect than the heterozygote. Since we've been doing a lot more research on ovine genomics, there are numerous breed-specific mutations that have been identified on a total of 4 different genes across the sheep genome. All of these have different gene actions, and it is, I want to say versigtig, you must be careful with regards to the type of mutation you can have, because some of them may actually lead to some sort of complete infertility. So you will see the breeds here. Those produce a fertile heterozygote and a very most fertile homozygote. But the third one, you'll see that the heterozygote, so it carries the mutation, but it also carries the wild type, is fertile, but if it carries 2 copies of the mutation, it becomes infertile. So depending on the mutation type and the gene within it is working, you get a different phenotype that is observed. Moving on to the infertility haplotypes. These are recessive mutations that cause embryo or fetal reabsorption. We just had Danuta speak about the fact that 10% of the embryos or the fetuses are aborted between 28 and 60 days, but actually 40 to 50% of fetal loss occurs before 42 days. These haplotypes, they act in a recessive fashion. They are widely known in the dairy industry. Those are just some of them. They are well known as the Holstein haplotypes, the Jersey haplotypes, and the brown Swiss haplotypes. And new research has actually identified some of these haplotypes in beef cattle, novel haplotypes. They are not these ones that are observed. The breeds that they have been seen in include the Limousin and the Simmentaler, so it is potentially going to be in other beef breeds as well. If you just look at my graph that I've made there, you will never have an animal that carries 2 copies of this infertility haplotype. If the embryo receives the infertility haplotype from the sire and the dam, that fetus is reabsorbed and the pregnancy is terminated. So it might not be that your animal can be in the best condition, you synchronize her, you give her the best semen, but are you sure that they do not carry these haplotypes that can cause fetal reabsorption? So the number one thing is to ensure that your bull that you use is not a carrier. Use clean bulls from these infertility haplotypes. Moving over to double muscling. Some of you might be thinking, why am I talking about double muscling? Well, it has an effect on fertility. We know that there are different mutations within the myostatin gene that cause double muscling. The 3 main variants present in South Africa are N2A21, QT04X, and F94L. The first 2 break the myostatin gene, rendering its function pretty useless, and muscles can grow as much as they want. The F94L mutation just changes the shape of the protein, so it does not bind as effectively to the muscle cells, so the muscle cells can still grow, but not as to the extent of NTA21 and QT04X. The reason that they are present in quite a few breeds in South Africa is that they were introduced through the upgrading system from the breeds that are known to carry them. And there are also a lot of many mutations with small to no effects. You'll see those at the top, those with the green. They don't really have an effect, but we observe that there is variation within the myostatin gene at these points. All those that are red, those are the bad ones, and the blue ones can be seen as kind of like F94L. So how does double muscling act? Well, it acts recessively. Carriers hide in the herd. You will see this animal is actually a carrier, the one in the middle. That animal carries QT04X, and what we have observed with QT04X is sometimes it shows incomplete penetrance. So an animal that is a carrier can either be observed as normal, or you can actually observe that the effect of double muscling. And that could be some form of epigenetic effect of QT04X switching off the myostatin gene on the other DNA strand. If you do mate 2 carriers together, you have a 25% chance of getting a double-muscled calf. The biggest effect is high birth weight. See here, that calf was stillborn. I've had breeders indicate to me that they have had calves of up to 76 kilograms born at birth, which is not very good for the cow. The observed phenotype on an animal that may survive past birth is the cylindrical body shape, that vorsi. You'll see on that animal on the left. What we see as well is the effect on fertility. Because the muscles are growing uncontrollably, it is moving all the energy away from fat, bone, and organ development towards muscle development. The animal doesn't have a lot of fat. Fat is the main signal for fertility. Fat produces a hormone called leptin, which then causes the gonadotrophic cascade to occur and for puberty to occur. If the animal doesn't have enough fat, it doesn't produce enough leptin, and therefore the reproductive organs cannot develop properly. We see a lot of animals getting rejected at inspection because they have underdeveloped testes, underdeveloped vulvas, and underdeveloped udders. That is an indication that that animal is potentially a carrier of double muscling or is a double-muscled animal. So, and then obviously the double muscling is excessively muscled. So what are the cow herd problems? Well, a cow doesn't want to give birth to a 76 kg calf. So she's going to have birth difficulties, dystocia instances. I mentioned the lower fat reserves. This affects fertility. If an animal is in an environment with high stress, it will not perform. But if it is in a good environment with lots of feed, low stress, that animal will perform. But in a poor, extensive environment, which the majority of you are farming in, that animal will not perform. And at the end of the day, you have reduced longevity within your herd. Your cow doesn't have enough fat reserves to conceive annually and to stay within the herd for the designated time you hope she can. Okay, moving over to diseases and viruses. So scrapie virus is known as transmissible spongiform encephalopathy. Some of you guys know about mad cow disease, how it occurred in the UK in the 90s and had to slaughter all of those cattle. The same type of virus, or it's not a virus, it's a prion, the same type of disease occurs in sheep and goats. And we can call it mad sheep disease or crazy goat disease, but it is caused by a prion. A prion is a rogue protein. Most of the proteins in your body facilitate you on your daily basis. They help you achieve homeostasis and do your things. But this protein is evil, and it can turn the other proteins evil. And what it does is, once it gathers a large enough group of fellow prions, they accumulate in the brain cells and they actually damage the neurons. So the animal actually gets twitchy, it feels scratchy. You can see that that sheep has scratched itself against a post or something to alleviate the symptoms it is feeling, but it is causing the animal to basically fade away. It is highly transmissible, especially through the placenta. It hasn't been detected in South Africa since 1972, but it has a long incubation period of 2 to 6 minutes. 2 to 6 years, I apologize. Meaning that you may not see the scrapie in your herd because most of your animals are going out of the herd before the incubation period is over. And there is currently no tests being done to see if scrapie is present in South Africa. The other one is lentivirus. Lentivirus is endemic to the Northern Hemisphere. But we know with globalization and the animals moving between countries that it may potentially make its way here. It's already in Ethiopia and in Nigeria. What is lentivirus? It's a retrovirus. Belongs to the same family as HIV, but it doesn't attack your immune system, it attacks your lungs. And the sheep basically get a form of pneumonia and they die because they can't absorb enough oxygen to maintain themselves. There is currently no treatment or vaccine for this. And there have been no tests done in South Africa. So we actually don't know if lentivirus is actually present in our sheep breeds. So why am I talking about scrapie and lentivirus? Because we can select for genetic resilience. We want to safeguard the biosecurity of our national flocks and herds. And the best way to do that is to improve the genetic resistance. For scrapie, there are haplotypes. We want ARR, we don't want VRQ. VRQ is the haplotype that causes the protein to become evil. We want ARR, that shows the highest resistance. Lentivirus, there's also been an E35K mutation observed. If they carry the K, they are basically immune to the lentivirus. They say it has to do with the biological mechanisms within that gene that lentivirus uses to make more versions of itself. So it affects that gene. My last point is detrimental mutations. These have a recessive mode of action. So carriers of certain disorders do not express the disease in any shape or form. I see it cut my slide. So animals could be mistaken as weak calves or poor calves when in fact they carry a genetic disorder. So we can split them up into metabolic and physical deformities. We spoke about glycogen storage diseases. Those come from Brahman-type animals or composite breeds that have used Brahman in the upgrading system. We've got BLAD, we know in the dairy, dumps, RDMS, McArdle syndrome, which is this picture, that occurs in sheep. They do not have glycogen phosphorylase, so they cannot convert glycogen to glucose. Physical deformities, we know BSD, I see I'm out of time, cardiomyopathy, CVM, here's crooked tail syndrome or Kromstert, hypertrichosis, which is a loss of hair, not to be confused with the slick gene of the Creole breeds. We also have SMA, that's the spinal something atrophy, I can't remember all of them, and mulefoot. All of these detrimental mutations can be tested for in a genomic test. So the genomic test, it's an all-in-one DNA test. You must ensure that your herd, your flock, your kennel of animals are free from any of these unwanted mutations. Make use of a genomic test to cover all of your bases. Recent research has actually shown that you can do a genomic test to detect aneuploidy or polyploidy within the animal. That means, does it carry only one chromosome instead of the normal 2 sets, or potentially more sets of chromosomes? Those can be done through copy number variants using a genomic test. So you don't need to do a karyotype anymore. You just do a genomic test. Other species available to test for disease, I basically mentioned cattle, sheep, and goats. We can also do this on horse breeds and dog breeds. The dog breeds, we know there are a lot of breed-specific ones, so they only go for those specific diagnostic tests. But the genomic test will allow them to cover all their bases. Make sure these dogs don't carry anything, even the horses. So let's make sure that our animals are genetically resistant to diseases and viruses that, as I said earlier, will improve the biosecurity of our South African herd and flock. My name's Jason. Thank you.